Now I request our Dean, Professor Anand K. Kondapi, to kindly introduce the Vice Chancellor, B.J. Roy, for his talk.
It's an honor to introduce Professor B.J. Rao Garu, our Vice Chancellor. He did his PhD from Usmania. Is it Usmania? ISC. Sorry, MSc from Usmania and PhD from Usmania. No, sorry, ISC Bangalore. Then he did postdoc in USA. And I know his work on recombination, where he worked in CM Rading's lab. And then he returned to India, worked at TIA for Bombay, then followed by he joined in ICER. And then from ICER, he is here with us as a Vice Chancellor last 4 years. And he's covered several areas of research, from bacteria to cancer research to plant sciences. You can listen to that, his research. So he's a vast experience, vast coverage in the area. And then he's very experienced and highly regarded in the science in India. And I request Professor B.J. Rao Garu to give his talk.
Thank you. Good morning to all of you. My warm welcome to Professor Takahashi. Professor Takahashi is, of course, an authority in photosystems. So obviously, I will not be talking about photosystems. Okay. Online. Yeah. Okay. So I also want to welcome very warmly all the participants offline here, and maybe there are some online participants also. GYAN is a very important national program. As Professor Rajgopal said, these are competitive programs in which we invite experts and let our participants go through an immersive experience by the teachings of the experts. This is a nationwide program and it's happening very, very well. So you are all very lucky that you are part of this learning experience. Chloroplast biogenesis, chloroplast physiology, of course, is an exquisitely evolved system. It's a beautiful system. Takahashi Garu knows the intricate details of it, but I want to tell a little bit about the physiology of it. I want to share some important aspects which are generally not well covered when we discuss about chloroplasts, especially the cell biology of chloroplasts. Chloroplasts are extremely well evolved adaptive entities, and they now are in constant coordination, homeostasis with other organelles, especially mitochondria, because mitochondria also is one of the most redox reactive organelle. Mitochondria is sitting here, and he has come for chloroplast, means there is an intense regulation between the organelles. And that's my first slide, actually. You can see that this is that's the mitochondria, this is the chloroplast. And most important aspect of these two organelles is the metabolites that they exchange as a function of different set of adaptive conditions. We still do not know the complete biology of those exchanges. That's number one. Number two, both these organelles are extremely active in retrograde signaling to the nuclear compartment. And in the retrograde signaling also, they coordinate with each other. They don't conflict with each other by and large. So therefore, that's a very important area of activity which a lot of plant physiologists are working on. So I'll touch upon some areas which I like best, need not necessarily of your interest, but I think these are the areas that I want to just very briefly mention, not going into too many details. How did chloroplast actually evolve? Was it an accident? People say it's an accident, but may or may not have. So you see that very interesting changes happen in the chloroplast, because chloroplast evolved when a photosynthetic organelle got engulfed by a eukaryotic cell. That was a fundamental event. Minus that, we wouldn't have been here. right? So when that got engulfed by a eukaryotic cell, obviously it has to go through a lot of adaptive changes. It's a bacterium that got engulfed. So bacterial cell wall has to be remodeled into chloroplast membrane. So many things have happened there. Phycobilisomes, peptidoglycan layer located between two membranes of cyanobacterial cell that got converted into the double membrane of the chloroplast. A very important adaptive change that happened when the bacteria got metamorphosed into an organelle. There are a lot of details available. I think it's a very fascinating area that one has to look into. Chloroplast is an exquisite organelle that is light sensing. All of you know it. But the chloroplast also leads its life in dark. We will discuss that a little bit later. So when the plant cells are exposed to light, differentiation of protoplast to chloroplast via intermediate etioplast. When etiolated, I'm reading this because reading is important to convey certain points. Etiolated seedlings sense light and switch to photomorphogenesis. There is activation of photoreceptors which activates photosynthesis associated nuclear genes. This is the heart of the issue, because a lot of nuclear genes are photosynthetically relevant, and these genes are activated when light is switched on, and this is done by undoing negative regulation. Who are the negative regulators? There are negative regulators here which are undone by inactivating negative regulators of photomorphogenesis like PIFs, COP1, SPA, etc., etc. This list is not exhaustive. So this is a very important trigger point when light is switched on, activating nuclear genes related to photosynthesis. I really want to read this text, if you don't mind. Chloroplasts are the organelles where oxygenic photosynthesis occurs and are therefore the enablers of autotrophy in plants. Beyond this well-known biological function, chloroplasts can also produce an array of pro-defense signals, including calcium waves, reactive oxygen species, and defense phytohormones such as SA and JA, salicylic acid and jasmonic acid. In recent years, accumulating evidence has indicated that chloroplasts not only function as environmental sensors and central signaling hubs. This is the most important part. It's a very intense signaling hub, but also acts as immunity-related motile organelles at infection sites. This area has to really become much more active now. A connection between plasma membrane and chloroplast during plant immunity mediated by protein trafficking has also been described. Meanwhile, chloroplasts can communicate with the nucleus through retrograde signaling pathway. This has been very well worked out, but the story is still evolving, which relays developmental and environmental cues and controls the expressions of thousands of nuclear genes and nuclear encoded chloroplast targeted genes. So this essentially sums up very important functions of chloroplast. Chloroplast stromule formation functions in plant immunity. I want to emphasize, my youngsters, that plant immunity is a very important area that you should look at in the context of chloroplast functions. Stromules are thin tubular extensions of the chloroplast formed in response to certain pathogenic stimuli that are believed to mediate the movement of chloroplast. Movement of chloroplast is very important. These are directed movements towards the nucleus in the initiation of immunity. This cell biology is a fascinating cell biology. I want all of you to really get into in future, if you can. Although findings over the past decade demonstrate that stromules are induced due to plant defense activation pathways and play a role in cell death, the question of whether stromules are directly required for immunity remains unanswered even now. Advances in this field were hampered by the lack of genetic systems. The genetics of stromule biogenesis is just becoming uncovered now, so right area to get involved into. Chloroplasts are key hubs for plant signaling. acting as environmental sensors and communicating with the nucleus via diverse mechanisms. Chloroplasts also play an essential role in plant immunity, while effectors from various pathogens have evolved to target chloroplast-mediated immunity either directly or indirectly. Despite that, our understanding of the role of chloroplast in plant defenses has deepened in the recent years. Plethora of questions await further investigations. I am reading these sentences because to me these sentences make a lot of sense. And I want to communicate more accurately the import of these sentences. For example, are all chloroplasts in a cell involved in mediating effective immunity? Is there a lot of diversity within the chloroplast community within a cell? Unanswered still. Are chloroplast-derived chemicals actively translocated to the nucleus during defense responses? If so, who are they? Do all compatible pathogens suppress chloroplast immunity? Very important question in the context of pathology of infections in plants. Answers to these questions will not only contribute to our understanding of chloroplast biology, but also propel the fields of plant immunity and plant pathogen interactions forward, potentially laying the foundations for generation of pathogen-resistant crops, which is what we need. Thus, decreasing crop losses, we certainly need this, and improving the crop productivity in the future. So very important problem areas get opened up. This is a very simple cartoon diagram. I'm sure in the GAN program you probably will discuss this. I've already discussed about it. It's a very important protein pathway where chloroplast, by its design, has transferred most of its important genes to the nuclear compartment during evolution. So therefore, it has to import the nuclear encoded proteins into the chloroplast. That is what is that nuclear gene transcription is under heavy control. And then protein import happens. And of course, chloroplast generates its own protein products. So a coordination between the translational products within the chloroplast and the imported translational products from cytoplasm, this coordination is exquisite coordination that generates lots of photosynthetic complexes that Takahashi Garu has been studying. So it's a very intimate coordination. No one can go out of stoichiometric range, right? So there is a lot of that, and there is very intense retrograde signaling to instruct the nuclear genome at the transcriptional level, how much to be produced at what time and how not to produce, how much is to be prevented, etc., etc. So that I'm sure all the details you will learn in the GAN program. This slide I really like. There is a multi-level reprogramming of gene expression. What do I mean by that? Why do we need this? We need this because sometimes the coordination is not so exquisitely modulated. Therefore, some proteins which are to be imported into the chloroplast, nuclear encoded proteins which have to be transported into the chloroplast, accumulate because the stoichiometric problems arise. So when these proteins that have to be imported into the chloroplast accumulate in the cytoplasm, that is not a good sign. That gives rise to a protein homeostasis response. And who is communicating this response? Most importantly, it is communicated by the ROS of the chloroplast to the nuclear genome. So, reactive oxygen species act as retrograde signal for the transcriptional activation of small HSPs, because there is protein homeostasis requirement now. Proteins have accumulated, they are getting deranged, therefore HSPs are required, chaperones are required, and a critical balance between HSPs, chaperones in response to retrograde signaling of ROS from chloroplast to the nucleus will alleviate the accumulation of misfolded proteins in the cytoplasm. Original destination of these proteins would have been chloroplast. So this is an important area, which is how stoichiometric ratios of multi-protein complexes in the chloroplast are actually maintained. This slide is very dense. I would rather go to the diagram and put down some important points here. This is very well described slide in the literature. But 2 points I have to mention. Chloroplast and mitochondria are intensely active in terms of generating electrons. And these electrons are extremely well directed from one carrier to the other carrier to the other carrier. But electrons being what they are, they can diffuse out. Whenever electrons get diffused out, one of the important consequences would be generation of reactive oxygen species. And who generates reactive oxygen species? The reduction of oxygen generates multiple forms of reactive oxygen species. Most safest form of reduced form of oxygen is what? Water. Ultimately, oxygen has to be reduced to water. Then there is no problem. But in the process of this reduction, multiple species accumulate. Some of them have a longer shelf life, which is what is the problem. But plants have converted those so-called harmful signals into useful signals. In this, there is a lot of reactive oxygen species that is getting generated, importantly at the leading end of photosystem 2 as well as at the lagging end. This is the end where electrons are received by PS2, and that's the end where electrons are given away by PS2. Can you just shrink it? I touched it, it enlarged. Okay, good. 2 important points I want to make. Generation of H2O2 at the front end of PS2, and that gets dismuted into superoxide here. And at the leading end, you have 2 electron flows generating superoxide radical H2O2 and most important hydroxyl radical. So PS2 is an intense generation of reactive oxygen species. But at the end, it is the reduction power that generates NADPH, most useful component, and generates ATP through the ATP synthase. These 2 are important products that lead into Kelvin-Benson cycle. My request to all the students here is to understand how different reactive oxygen species are generated here in the process of electron flow. The previous slide actually had a lot of details which I have not gone through. I request all of you, my students, to go through those details. If you look at this, there are 2 important components I have put down. One is tryptophan, another is thiol containing residues. These 2 are exquisite chemical entities in capturing the redox signals. Tryptophan goes into multiple states: indolyl radicals, hydroperoxide. You can see the chemistry changes exquisitely. I do not have time to go into those details, but multiple forms of tryptophan redox states exist. And all these happen on the protein surfaces, not as pre-tryptophan. We still do not understand different states of tryptophan redox states as a function of protein complexes in chloroplast systems. Similarly, you have thiol residues which go through a lot of changes: persulfide, S-nitrosothiol, S-glutathione, sulfenic acid, and disulfide. So multiple forms of these redox states in these 2 chemical entities on proteins as a function of different conditions is a way of sensing the redox state of that organelle. And this in turn would lead to gene expression modulation at the nuclear compartment. There is a lot of biochemistry that goes on here whose intricate details we still do not have a full details of. Now this, I just want to summarize a little bit. Neither chloroplast nor mitochondria are static entities. They modulate with each other. Some of these things we looked at. Gunjan, my student, is sitting here. Gunjan essentially looked at how these two compartments in Chlamydomonas reinhardti coordinate as a function of diurnal cycle. You know the diurnal cycle, 12-hour light, 12-hour dark. Is there any coordination between the output of these two organelles? And Chlamydomonas reinhardti is a very good system to study because there is a single chloroplast, cup-shaped, in apposition of which is the mitochondrial compartment, in physical apposition of which. So we measured the output of both these organelles as a function of diurnal cycle, 12-hour light and 12-hour dark in a time course series. And what we observed, which is published recently, is this. Both photosynthetic activity as well as the mitochondrial activity as a function of time during daytime increase, the output increases, and as a function of time in the dark phase, the output drops. Concordantly, both organelles actually go concordantly with each other. We measured a lot of photosynthetic and mitochondrial activities. They go hand in hand together in light and dark dependent manner. We still do not know whether there is a mutual coordination between them. However, when we study exactly the same thing in TOR kinase mutant cells, the trend is observed, but the trend is suppressed significantly. Both these oscillations, output oscillations, get dampened in TOR kinase mutant system. We do not know the molecular mechanism. We're trying to look at it. Perhaps the metabolic signals feed into these and try to give the level of regulation that the system requires. You can see that the mitochondria gets fragmented very heavily in the dark, whereas the same mitochondria in the light phase is exquisitely reticulated. It is much more reticulated in the light phase. You can see that. And both chloroplast and mitochondria really physically oppose with each other. This is a GFP tagged mitochondria and autofluorescence managed chloroplast. So both are studied in the live system. So there is a physical apposition. They coordinate with each other's functions. So I want to sort of conclude here. I wanted to communicate to you two important points. And one is that I want my youngsters to look at chloroplast as an adaptive signaling hub and a very important organelle for plant immunity. This is one point that I want to make. The other point is, do not look at chloroplast biology in its isolation. Look at it in the context of mitochondrial functions, look at it in the context of nuclear functions, etc., so that you have a relatively better appreciation of chloroplast compartment in concert with overall cell biology of the system. These are the 2 points I wanted to communicate. I think some of these slides did communicate that, but do stay with chloroplasts. Very important organelle, and bring in mitochondria where you can. Thank you.
Thank you so much, sir, for this great talk. I'm sure all the audience members have been left inquisitive with all the unanswered questions with this talk. So now, as we draw the inaugural session to a close, I invite Professor Rajagopal once again for the vote of thanks.
Yeah, sir, thank you for an excellent talk. So I agree that, both this chloroplast is not really isolated, both mitochondria, not only mitochondria, many other things. There is communication between chloroplast and mitochondria and cytoplasm, where we have been discussing with Naresh. You should collaborate with chloroplast with mitochondria. I think it will happen soon.
He smiles but doesn't help.
So thank you so much for your really eye-opening the discoveries also. Many discoveries have happening. As I said, now the photosynthesis is not only for food. It is have many other applications, biological, medical applications. So many things. You can do a lot of engineering in chloroplast and do a lot of even the people are using for now biotechnology application, energy applications, hydrogen production. and several other things that we'll discuss in the class. So there are 30 participants now from Orissa to Kerala from north. It's not only located only to the Hyderabad. Many are from different parts of India. So and also happy to say that I'm conducting the 4th GIAN meeting. So it's almost like 16, 17, 18, and then now 25. I hope, but it was very difficult to conduct these kind of meetings, coordinating with the funding body, university, with all participants. So anyway, thank you all for
the best
joining.
part is to invite people like him.
Yeah.
That's the best part.
Yeah. So now I would like to thank our again Vice Chancellor for giving his time and giving an excellent talk where the interaction between chloroplast and mitochondria, where also put his some of his research work. And also, again, I would like to thank Professor Echiro Takahashi for coming all the way and spending with us. And you will hear all the talks on photosynthesis and engineering. And Dean School of Life Science, Anand Kondepi Garu, and head of the department, and faculty colleagues, participants, and other students, my own students, those who are actively involved. And also recently, we contacted some Professor Tripathi is joining for the biogenesis, chloroplast biogenesis. He is going to give a few more talks, and other resource persons. I would like to thank everyone. And finally, I would like to thank MHRD for sanctioning this GIAN, and also our finance department. And finally, I would like to thank Green Tech and Cytiva and INCARP for giving small money for conducting this meeting. Thanks for all of you for joining this inauguration session. Thank you.